Hello, dear students. Today we're going to speak about inheritance, variation. Let's start. The Earth is inhabited by millions of organisms, every one of which is unique. Individuals belonging to different species are usually easy to distinguish. Members of the same species may differ only in small ways. But even clones show some subtle differences. The differences between individuals of the same species are called variation. These differences may be the result of genetic differences, the influence of the environment, or a combination of genetic and environmental influences. Genetic variation. Genetic differences reflect the genotype of an organism, that is, its genetic makeup. A diploid organism has two sets of chromosomes and two forms of each particular gene. These alleles may be the same or different. If different, one of the alleles may mask the other allele. The dominant allele is therefore expressed in either the heterozygous or the homozygous condition, whereas the recessive allele is expressed only in the homozygous condition. If an organism is haploid, all its alleles will be expressed and will be reflected in its observable or measurable characters. Phenotypic variation, continuous and discontinuous. The measurable physical and biochemical characteristics of an organism, whether observable or not, make up its phenotype. The phenotype results from the interaction of the genotype and the environment. The genotype determines the potential of an organism, whereas the environmental factors to which it is exposed determine to what extent this potential is fulfilled. For example, in humans, the potential height of a person is genetically determined, but a person cannot reach this height without an adequate diet. Phenotypic variation is of two main types, continuous and discontinuous. In continuous variation, differences are slight and grade into each other. Characteristics such as human height and weight show continuous variation and are usually determined by a large number of genes and considerable environmental influence. In discontinuous variation, the differences are discrete and clear-cut. They do not merge into each other. Discontinuous variations are generally caused by different alleles of one, two, or only a few genes. Continuous variations are usually quantitative, whereas discontinuous variations are qualitative. Thus, height in humans is a continuous variation given a value in meters, whereas height in sweet peas is a discontinuous variation described as tall or dwarf. Mutations, more variation. Genetic variation arises partly from sexual reproduction by a combination of independent assortment, crossing over, and random fertilization. However, these processes merely shuffle the existing pack of genes so that new combinations are made. The ultimate source of inherited variations is mutations. A mutation is a change in the amount or the chemical structure of DNA. If the information contained within the mutated DNA is expressed it can cause a change in the characteristics of an individual cell or an organism. Mutations in the gametes of multicellular organisms can be inherited by offspring. Mutations of the body cells of multicellular organisms are confined to the body cells derived from the mutated cell. They are not inherited. Mutations can happen spontaneously as a result of errors in DNA replication or errors during cell division, or they can be induced by various environmental factors, such as certain chemicals, X-rays, and viral infection. Factors that induce mutations are called mutagens. Chromosome mutations and gene mutations. Alterations in the number or structure of chromosomes are called chromosome mutations. Chromosome mutations can happen during mitosis and meiosis when chromosomes are being condensed and pulled apart. Homologous chromosomes may fail to separate, resulting in non-disjunction. Chromosome mutations also occur during interphase when DNA replicates and during crossing over when sections of chromosomes are interchanged. Gene mutations are changes in the nucleotide base sequence in a cistron. A change of a single nucleotide base pair is called a point mutation. There are a number of types of point mutation, including substitution, the replacement of one nucleotide with another containing a different base; deletion, the loss of a nucleotide; insertion or addition of an extra nucleotide. Real-life example: sickle cell anemia. Sickle cell anemia is an example of an inherited condition that results from a substitution. Gene mutations may also result from duplication and inversion. That is the end of our video session. Thank you for watching.